Assalamu alaikum. Myself am Rasafannisa from second year MSc Microbiology from Sacred Heart College. Today my topic is about food preservation. As we all know, food is the most fundamental requirement for our human life, even than our shelters and our clothing. So, let me discuss how to preserve the food for our future use. Let's see what is food preservation. Food preservation is essential for extending the shelf life of food products, preventing spoilage, and ensuring the safety from foodborne illnesses. There are various methods are employed to inhibit the microbial growth, maintain food quality, and enhance the longevity of food items. There are different types of food preservation, which includes high temperature preservation, low temperature preservation, high pressure preservation, drying, aseptic packaging, pulsed electric feed, which is also known as PEF, modified atmosphere packaging, which is also known as MAP. Let's see what is high temperature preservation. In high temperature preservation methods, which involves the heating of a food to kill or inactivate the harmful microorganisms and enzymes that cause spoilage. This can be achieved through several techniques, which includes pasteurization, canning, and sterilization. Pasteurization. This method involves heating liquids like milk to a specific temperature for a set period to kill pathogen without altering the food qualities significantly. Common pasteurization temperatures range from 60 degrees Celsius to 85 degrees Celsius. As we all know that this method was discovered by Louis Pasteur. And next, now about it canning. What is canning? This process seals food in an airtight container and heats them to high temperature, typically above 100 degrees Celsius, to destroy the bacteria and spores. Canning is widely used for fruits, vegetables, meats, and soups. Sterilization. This method involves heating food at temperature above 100 degrees Celsius for longer periods to ensure complete destruction of all microorganisms. It is commonly used in the production of shelf stable foods. Now let's see what its advantages. It is effective in killing pathogen, it extends the shelf life significantly, and it also maintains the nutritional value when done correctly. Now about low temperature preservation. In low temperature preservation techniques primarily involve refrigeration and freezing to slow down the microbial growth and enzymatic reactions. Refrigeration. Keeping food at temperature between 0 degrees Celsius and 4 degrees Celsius slow down the growth of most bacteria. This method is ideal for short-term storage of perishable items like dairy products, meats, and fresh produce. In freezing method, freezing food at temperature below 18 degrees Celsius halt microbial activity entirely. While it preserves the nutritional quality of food effectively, it can affect texture upon thawing. Now let's see about its advantages. It is simple and convenient for home use. It maintains quality over extended periods. It also reduces spoilage rates significantly. High pressure preservation. In high pressure processing involves subjecting food to pressures exceeding 100 MPa megapascals. This technique disturbs the microbial cell membrane without the need for high temperature. Now let's see about its mechanisms. HPP inactivates the bacteria, yeast, and mold while preserving the sensory quality of the food. It is often used for juice, ready to eat meals, and daily meats. About its advantages, it retains the flavor and nutritional value. It extends the shelf life without preservatives. It also reduces the need for heat treatment. Drying. It is also one of the type of the food preservation. In drying, there are different types of drying, which includes air drying, freeze drying, and sun drying. In air drying, it involves the circulating of warm air around the food. In freeze drying, also known as lyophilization, it involves the freezing the food and then reducing pressure to allow ice to sublimate directly into vapor. In sun drying, it utilizes solar energy but it is weather dependent. Radiation. It is also one of the most widely used technique for food preservation. Radiation preservation employs ionizing radiation, which includes gamma rays, X rays, to eliminate the microorganisms. About its mechanism, radiation disturbs the DNA in bacteria and parasites, preventing them from reproducing. It is commonly used for sterilizing spices, dried fruits, meat products, and some vegetables. Modified atmosphere packaging, which is also known as MAP. MAP involves altering the composition of gases surrounding packed foods. Its mechanism is typically reduce the oxygen level while increasing the carbon dioxide or nitrogen level to slow down the oxidation and microbial growth. This method is often used for fresh produce, meats, and baked goods. Let's see another type of the food preservation, which is pulsed electric field. As the name, we can able to know that this method involves electric field to preserve the food. Let's see its definition. PEF technology use short bursts of high voltage to create pore in microbial cell membrane. About its mechanism, the electric fields disturb the cellular integrity without significant heat application. It is particularly effective for liquid foods like juice and soups. About its advantages, it retains the flavor, color, and nutrients. It also reduces the processing time compared to traditional method. It environmentally friendly with lower energy consumption. Aseptic packaging. Aseptic packaging combines the sterilization process with packaging techniques.